Hi everyone. Have you ever seen the beautiful colors of a peacock feather, soap bubbles, or birds that seem to change color as they move? Have you ever wondered what color is, or why some animals, birds, and natural objects are so vibrant while others appear dull or colorless? Today, we are stepping into the intersection of biology, physics, and material science. We often perceive the natural world through a lens of chemical pigments, chlorophyll in leaves or melanin in skin. However, nature has spent millions of years mastering a different kind of coloration, one that uses physics instead of chemistry. This is the world of structural coloration and natural photonic crystals. In this lecture, I will talk about how and why living organisms and natural objects manipulate light. We will explore the origins of color in nature, beginning with pigmented colors using selected molecules and polymers, and then moving to a fascinating alternative, structural color, which arises from optical nanostructures. Finally, we will look into scattering and biological transparency. This lecture is designed for an interdisciplinary audience and for anyone curious about the magic of colors in nature. The concepts are presented in a simplified and intuitive manner. Color plays an important role in our daily lives. It helps us recognize and remember objects, evoke emotions, and even influence decisions such as purchasing behavior. Humans perceive color because our eyes are sensitive to the visible region of the electromagnetic spectrum, roughly between ultraviolet and infrared light. Although humans cannot see ultraviolet or infrared radiation, certain organisms, such as bees, can perceive ultraviolet light, and snakes can detect infrared radiation due to differences in their eye anatomy and specialized pit organs. The ability of living organisms to perceive different colors has implications for pollination, prey capture, and food identification. Therefore, perception of color has evolutionary significance. Our ability to distinguish colors and brightness depends on the complex architecture of the human eye, which contains specialized photoreceptors called rods and cones. Rods are responsible for detecting brightness and intensity, while cones are responsible for color vision. Humans typically have three types of cones sensitive to red, green, and blue light. Interestingly, about 12% of women may possess tetrachromatic vision, allowing them to perceive a wider range of colors. At the same time, color blindness, most commonly red-green color blindness, highlights that color perception varies across populations. One fascinating question is, can colors be generated from colorless materials? Here is a video demonstrating dynamic color change on a silicon wafer coated with a very thin and completely colorless polymer film. When exposed to moisture, the surface displays vivid colors. When warm air is applied, the colors disappear. However, the colors reappear when moisture returns. This is an example of dynamic color manipulation in response to environmental conditions. What is remarkable here is that no pigments are involved. The material itself is colorless. This type of dynamic color change is fascinating and motivates us to ask fundamental questions. What is color? How is color produced? Why does nature use color so effectively? In nature, dynamic color change is often used for defense, camouflage, communication, and environmental adaptation. Living organisms manipulate light through several physical processes, including absorption, reflection, scattering, diffraction, interference, transmission, waveguiding, and lensing. What is extraordinary is that biological materials are made from simple and common substances, yet they display remarkably precise optical engineering. Nature achieves this using minimal resources, high efficiency, and structures that often perform multiple functions simultaneously. All matter absorbs light, but color arises only when absorption occurs at specific wavelengths. Without selective absorption, there would be no color, no vision, no photosynthesis, no thermal regulation, and no biomass production. For example, DNA absorbs strongly in the ultraviolet region between 240 and 275 nanometers due to electronic transitions in its nucleotide bases. Proteins also absorb ultraviolet light depending on their amino acid composition. Carotenoids and metal complexes absorb visible light and produce vivid pigmentation. Water preferentially absorbs infrared radiation, contributing to heat generation and temperature regulation in biological tissues. However, ultraviolet radiation can damage DNA and cause molecular damage. Nature addresses this problem by using sacrificial UV-absorbing pigments such as melanin and carotenoids that protect essential biomolecules. Let us look at some examples of pigmented colors before moving to structural colors. Carotenoids are particularly remarkable molecules. They have high absorption coefficients, function as light harvesting molecules, provide UV protection, and contribute to biological coloration. A well-known example is the Flamingo, whose pink coloration depends on carotenoid-rich shrimp in its diet. When this dietary source is absent, the color fades, illustrating a key characteristic of pigmented color. Another striking example of pigment-based functionality is Octopus Ink. When threatened, an octopus releases a dense cloud of ink composed largely of melanin. This ink absorbs light across ultraviolet and visible wavelengths, forming a thick, smoke-like cloud that confuses predators and enables escape. Melanin is a complex natural polymer. Its exact molecular structure is still not fully known, and it cannot yet be perfectly synthesized. Beyond coloration, melanin provides UV protection, mechanical strength, metal ion binding, and enhanced virulence in certain microorganisms. This demonstrates that color in nature is not merely aesthetic but closely linked to survival. We now move to the central topic of this lecture, that is the structural color. Structural color arises not from pigments but from the interaction of light with nanoscale structures. In 2005, an intriguing question was raised. Why are male birds often more colorful than females? A classic example is the peacock, whose elaborate tail feathers play a critical role in mate selection. Experiments have shown that symmetry in the eye patterns of peacock feathers is essential for attracting mates. Structural color has therefore evolved not only for defense but also for communication and sexual selection. Structural colors originate from physical phenomena such as interference, diffraction, and selective reflection of light. These effects occur in structures known as photonic crystals or photonic bandgap materials. We encounter similar effects in everyday life, such as compact discs, soap bubbles, and oil films on water. Unlike pigmented colors, structural colors do not fade with time and often change with viewing angle. Then, what are photonic crystals? How do they create colors? Photonic crystals are periodic structures made of dielectric materials. They consist of periodically modulated structures of different dielectric materials with the periodic length nearly equal to the light wavelength. They can be one-dimensional, two-dimensional, or three-dimensional. These structures control the propagation of light in a manner analogous to how electronic bandgaps control electron motion in semiconductors. They create photonic bandgaps, forbidden energy ranges that block specific light wavelengths, allowing for precise light confinement, filtering, and enhanced optical, biosensing, and computing devices. Photonics crystals occur in nature, for example, butterfly wings, opals, where periodic structures cause iridescent colors through diffraction rather than pigments. In biological systems, photonic crystals are constructed from simple materials such as chitin, keratin, cellulose, guanine, and melanin. Their remarkable optical properties arise from nanoscale organization rather than chemical complexity. Photonics crystals can be classified by dimensionality into 1D, 2D, and 3D. One-dimensional photonic crystals, composed of alternating dielectric layers, thin films with different refractive indices. They are used in Bragg mirrors, anti-reflection coatings, and thermochromic sensors. Two-dimensional 2D photonic crystals feature a periodic refractive index in two directions, often created by patterning holes or rods in a substrate. Examples include photonic crystal fibers, PCF, which use air holes in the cladding to guide light, and slab structures. Three-dimensional 3D photonic crystals are periodic in all three directions, allowing for a complete photonic band gap. Examples include natural opals, synthetic opals, close-packed spheres, and inverse opals. One-dimensional photonic structures are among the most common in nature. A beautiful example is found in hibiscus petals. Although they appear pigmented, certain regions exhibit iridescent color that changes with viewing angle. In this specific example, petals are white with a patch of red pigment at the base. The pigmented patch is iridescent. The pigment appears blue, green, or yellow depending on the angle from which it is viewed. In many species, such as tulips, iridescence is only evident to humans when the pigment color and the petal surface structure are separated. Electron microscopy reveals diffraction gratings with periodicities comparable to the wavelength of visible light. Behavioral studies show that bumblebees preferentially recognize iridescent flowers over pigmented ones, demonstrating the biological significance of structural color in pollination. Using a bee hexagon color space model, researchers determined that the variation in hue from a single hibiscus trionum flower is sufficient for ready visual discrimination by bees. In differential conditioning experiments, bumblebees, Bombus terrestris, successfully learned to distinguish rewarding iridescent discs from non-rewarding non-iridescent discs, reaching over 80% accuracy after 80 visits. Dynamic structural color is also observed in insects such as the Japanese jewel beetle. This beetle displays multiple colors using the same materials with color variation arising solely from differences in layer thickness and number. When the beetle is at rest, its elytra, modified hardened forewings that cover the flexible transparent hindwings, reflect maximally in the green region. This green pattern is interrupted by longitudinal dark purple stripes, and the borders between the green and purple areas display red-orange coloration in the cuticle. The underside of the beetle is highly curved and uniformly colored orange. In all cases, an approximately 1.3 micrometer thick distal sheet forming the epicuticle is present and consists of several layers with alternating high and low electron density. The number of layers varies across color regions, with about 16 layers in the green area, 22 in the orange area, and 12 in the purple area. The layers are not discrete, but instead exhibit a graded structure, so the refractive index does not change abruptly from one layer to the next. Rather, the refractive index oscillates between approximately 1.6 and 1.7 with a periodicity that depends on the location. This spatial variation in refractive index gives rise to different reflectance spectra and, consequently, to the observed differences in color. Morpho butterfly wings provide another iconic example. Their intense blue coloration, visible from many angles, results from air chitin nanostructures that enhance reflectivity. The optical characteristics of the Morpho wing include very high reflectivity within a selective wavelength range, a uniform blue reflection over a wide angular range, and variations in color tone and gloss among different species. While a simple multilayer interference model can successfully explain the first characteristic, high reflectivity at specific wavelengths, it fails to account for the wide angle uniformity of the blue reflection or the interspecies variations in color and gloss. Some fish and beetles exhibit physiologically controlled color change by altering refractive indices through fluid movement, enabling rapid reversible camouflage. Neon tetras naturally change color based on light conditions and mood, often appearing pale or faint at night or upon waking and becoming vibrant blue-green in daylight. Their signature stripe can turn deep violet or dull when sleeping, stressed, or in poor water quality due to shifting guanine crystal platelets in their scales. Charidotella egregia, the golden tortoise beetle, reversibly alters its cuticle from metallic gold to a dull orange-red when stressed, using a moisture-dependent structural mechanism. The gold color, produced by a chirped multi-layer reflector, Bragg mirror, in the cuticle that reflects light via interference, is lost when the beetle expels water, transforming the structure into a translucent scattering layer. Two-dimensional photonic structures are found in organisms such as peacock feathers and sea mice. In peacock feathers, melanin rods arranged in different lattice geometries produce distinct colors using the same material. Compared with one-dimensional photonic nanostructures, two-dimensional structures are relatively uncommon. In nature, surfaces with 2D gratings are generally not designed to generate bright structural colors, but instead serve anti-reflective functions, increasing the efficiency of the animal's vision while reducing unwanted reflections that might attract predators. In addition, these 2D gratings can also exhibit self-cleaning capabilities, owing to the high fraction of air trapped in the trough regions between the arrays. Researchers have discovered and characterized a two-dimensional photonic nanostructure in the hairs of the sea mouse, which consists of periodic long fibers. Each sea mouse hair contains thousands of hollow, close-packed, longitudinally oriented cylinders with diameters of about 230 nanometers. Bright structural colors are produced when these cylinders collectively diffract light incident on the side of the hair fibers. Transmission electron microscopy (TEM) of a strongly reflecting red spine obtained by slicing the hair with a microtome into transverse sections reveals an array of hollow cylinders whose long axes run along the spine. The images correspond to the wall region of the spine, while the center remains hollow. The hollow cylinders have thicker walls near the edge of the spine to provide mechanical rigidity, after which the wall thickness decreases over a few layers to a constant value. The cylinders are close packed, with each cylinder surrounded by 6 nearest neighbors. The spacing between adjacent layers of cylinders is approximately 0.51 micrometers and remains constant throughout the wall cross-section, which contains about 88 layers of close-packed voids. The cylinder walls are composed of pure alpha chitin with a refractive index of about 1.54. Another fascinating example is the peacock feather. Each barbule contains a medular core of approximately 3 micrometers that is enclosed by a surrounding cortex layer. The cortex of all barbules hosts a two-dimensional array composed of melanin rods embedded in and connected by keratin, with each melanin rod having a length of about 0.7 micrometers. Despite the wide range of observed colors, the underlying structures in differently colored barbules are remarkably similar. In blue, green, and yellow barbules, the lattice formed by the melanin rods is nearly square, whereas in brown barbules, it adopts a rectangular lattice. The primary differences between these structures lie in the lattice constant corresponding to the spacing between the rods and in the number of periods, that is, the number of melanin rod layers along the direction normal to the cortex surface. Birds of paradise represent a unique and striking example of structural color evolution driven by sexual selection. Male birds of paradise captivate females through an extravagant dancing ritual known as the ballerina dance, during which their intensely colored feathers are prominently displayed. In contrast, females retain a dull brownish plumage close to the ancestral color state. Males are characterized by predominantly black feathers adorned with strongly silver-colored patterns arising from a unique boomerang-shaped feather construction enveloped in a thin film that produces three-directional reflections. As the bird moves, these feathers rapidly switch between orange, green, and blue hues. In Laws Parotia, the occipital, nape, and breast feathers exhibit distinct nanostructures. The barbules in both regions contain well-ordered layers of melanosomes embedded within a keratin cortex, but with markedly different morphologies. Occipital feather barbules are flat and contain melanosomes of approximately 250 plus or minus 20 nanometers in diameter, with an interlayer spacing of about 400 plus or minus 20 nanometers. In contrast, breast feather barbules contain much smaller melanosomes, around 120 plus or minus 15 nanometers in diameter, with an interlayer spacing of roughly 230 plus or minus 20 nanometers. In addition, the layers in the breast barbules are skewed, producing a boomerang-shaped cross-section. These markedly different barbule morphologies give rise to distinct spatial and spectral reflection properties. The feathers combine multi-layered pigmentary and structural coloration with quasi-ordered spongy arrangements of melanosomes, nano-sized melanin-containing granules that both absorb and structure light. Melanin not only contributes directly as a pigment, but also enhances, filters, and refines structural colors by absorbing incoherently scattered light, thereby increasing color purity. The precise self-assembled arrangement of melanin thus provides an efficient and versatile route for generating the remarkable diversity of colors seen in bird of paradise feathers. Three-dimensional photonic crystals are common in beetles and butterflies. These structures often resemble opals or complex gyroid networks and produce uniform metallic coloration. Beetles living in dimly lit tropical forests often display vivid structural colors which under direct sunlight may appear only as localized spots of light, since multi-layer reflectors on their rounded surfaces act like tiny mirrors. In most cases, only part of the insect is visible from any given direction. However, Pachyrhynchus argus exhibits a uniform metallic coloration that remains visible from all viewing angles. This omnidirectional coloration arises from a photonic crystal structure analogous to that found in opal. The color is produced by scales approximately 0.1 millimeters in diameter that occur in patches on the top and sides of the beetle's roughly hemispherical body. These flat scales lie parallel to the body surface and consist of two components, an outer shell and an inner structure. In some scales, the inner structure is absent, rendering the entire scale transparent. Scanning electron microscopy reveals that the internal particles are arranged in flat layers with a precise hexagonal close-packed order. When the lattice constant of this crystal approaches half the wavelength of visible light, the structure acts as a three-dimensional diffraction grating, forming optical domains within a single scale. This nanostructure is the most common packing arrangement found in opal, although opal microspheres may occasionally adopt face-centered cubic packing, and it enables the reflection of a narrow range of wavelengths over a wide range of incident angles. The visual appearance of the butterfly P. sesostris is particularly striking. While the overall color of the upper wing surface is jet black due to highly melanized scales, large green spots dominate the forewings and smaller red spots mark the hind wings. These colored regions arise from photonic crystals composed of assemblies with a gyroid-type morphology, with a chitin filling fraction of about 30 to 40% and a cubic unit cell size in the range of 280 to 330 nanometers. The photonic crystals are overlaid by a thick lamellar structure known as a honeycomb. Notably, other minimal surface-based photonic crystals such as simple cubic P-type or diamond-type D-type structures have not been observed in butterflies. In contrast, diamond-type photonic crystals have been identified in beetles, whereas gyroid-type photonic crystals have not yet been discovered in beetles, suggesting that the developmental pathways underlying photonic crystal formation differ fundamentally between insect groups. This observation also raises the question of whether different crystal types with distinct iridescent and polarization characteristics confer specific functional advantages to different insects. In Paradise species, the green and red wing patches are presumed to function as aposematic signals, as these butterflies are known to elicit aversive behavior in bird predators. More generally, many insect species exhibit green coloration for camouflage, which is most often produced by structural coloration, with only a few rare pigment-based exceptions. Helicoidal structures represent another fascinating class of optical architectures. These chiral structures selectively reflect circularly polarized light and enable communication and navigation in certain insects. Many species, including fish, cephalopods, crustaceans, and insects, have evolved the ability to perceive polarized light, using polarized vision for navigation, communication, and mate selection. Over the course of this evolutionary process, a variety of organisms have developed structural colors based on helical architectures. A defining characteristic of structural color generated by such helical structures is its strong angle dependence, which gives rise to striking iridescence and the selective reflection of single-handed polarized light. A prominent example is the exocuticle of the beetle Chrysina gloriosa, which selectively reflects left circularly polarized light and therefore exhibits a brilliant metallic appearance. When left circularly polarized light is removed from the incident illumination using a quarter-wave plate and a polarizer, the beetle's characteristic bright green reflection disappears. This selective reflection of circularly polarized light originates from nanoscale helicoidal features in the exoskeleton, whose periodicity is comparable to the wavelength of visible light. The fruits of Margaritaria nobilis possess a helicoidal cell wall architecture that generates an intense blue-green coloration. In the related case of P. condensata, the refractive index of dried cellulose is approximately 1.53, and the mean value of the helicoidal pitch P is about 290 nanometers. Using the relation presented in this equation and inserting the values mentioned above, yields a reflected wavelength of roughly 445 nanometers, corresponding to blue coloration. Remarkably, the reflectivity of the fruit reaches about 30% of that of a silver mirror, making it one of the highest reflectivities reported for any biological material. In addition, uniform nacreous aragonite layers contribute to multi-layer interference, while closely spaced surface grooves act as a reflection grating, producing a diffraction pattern. The resulting bright spots are diffuse rather than sharp, owing to the uneven microstructure of the grooves. The uniform nacreous layers of aragonite contribute to multi-layer interference effects, while the closely spaced surface grooves give rise to a diffraction pattern associated with the reflection grating structure of the shell. The resulting bright spots are diffuse rather than sharp, a consequence of the uneven microstructure of the grooves. Not all optical effects involve vivid color. Bright white coloration arises from multiple scattering in disordered nanostructures, while biological transparency, common in marine organisms, requires precise refractive index matching with water. We now turn to another optical phenomenon, scattering. While sub-micrometer thick films are sufficient to generate colors through interference effects, achieving a broadband white reflection is far more challenging because all wavelengths of visible light must be reflected with comparable efficiency. This typically requires much thicker films owing to the optical requirements for multiple light scattering. To produce uniform white coloration, nature therefore relies on scattering from disordered arrangements of randomly positioned scattering centers. Butterflies of the Pieridae family are a classic example, exhibiting bright white coloration that arises from strong scattering of incident light combined with absorption by pterin pigments within the scale structures. Pterins are a class of pigments with distinct absorption properties. Leucopterin absorbs primarily in the ultraviolet, while erythropterin absorbs up to wavelengths of about 500 nanometers. Ellipsoidal beads made of pterin enhance wing reflectance, by increasing brightness at wavelengths where pigment absorption is negligible. Correspondingly, white scales are densely covered with these ellipsoidal beads, whereas black scales lack them entirely, indicating a clear optical function for the hollow pterin beads. Certain beetles, such as Siphoculus and Lepidiota stigma, display an exceptionally bright whiteness resulting from pronounced multiple scattering. Unlike Pieridae butterflies, the beetle scales are among the most strongly scattering low refractive index materials known. In ordered assemblies, a lower refractive index of the scattering elements would normally require a thicker structure to achieve high brightness, reducing overall scattering efficiency. Remarkably, these beetles achieve extremely high scattering strength even with very thin scales due to the anisotropic shape and orientation of the scattering elements that form a disordered network. As a result, the scales appear white from all angles. Light is scattered within an internal random network of interconnecting rod-like chitin filaments, where disorder has evolved to suppress spatial correlations while maintaining a high filling fraction enabled by the anisotropic scatterer geometry. This anisotropic arrangement leads to anisotropic light transport, enhancing scattering along the out-of-plane direction and producing a pronounced total reflection, perceived as high brightness. Consequently, the degree of anisotropy emerges as a key microscopic structural feature governing bright white reflectance in thin, low refractive index optical coatings. Biological transparency represents the optical opposite of light absorption, in which light is transmitted almost entirely through a biological object or surface with minimal optical loss due to absorption or scattering. Maintaining transparency is challenging, as small changes in temperature or salinity can make organisms suddenly visible. Behavioral adaptations, such as living in deeper waters, help overcome this limitation. This property is significant because it provides effective camouflage and near invisibility from predation, while also reducing the metabolic energy required to synthesize colored pigments. For transparency to be effective, it must be maintained consistently throughout the entire volume of an animal's body, with the notable exceptions of the eyes and the gut. However, biological transparency is highly sensitive to environmental conditions, and organisms can lose their ability to maintain natural transparency when those conditions change, leading to increased visibility to predators. Grass shrimp, for example, are hypersensitive to variations in water salinity and temperature. Changes as small as about 5 parts per thousand in salinity or only a few degrees in temperature can reduce their observed transparency from roughly 40% to nearly zero. To cope with these challenges, such organisms have evolved adaptive behavioral responses, such as migrating to deeper water to avoid environmental conditions that would otherwise compromise their transparency. In conclusion, nature demonstrates extraordinary mastery in manipulating light. Color can arise from pigments, structural order, disorder, polarization, or transparency. 1, 2, and 3-dimensional nanostructures enable a wide range of optical effects that serve functions far beyond aesthetics, including survival, communication, camouflage, and reproduction. I hope this lecture inspires you to view colors differently and to ask whether a color is pigmented or structural. If you find this lecture useful, remember to like, comment, and share it with others.